Passive transport is the transport of molecules down its concentration gradient, which is from an area of a high solute concentration to an area of a low solute concentration until equilibrium is reached. Passive transport does not expand any energy from the cell. Equilibrium is when there is the same concentration of solutes inside the cell as in the solution surrounding the cell. Diffusion is the movement of molecules down its concentration gradient through a cellular membrane to achieve equilibrium. Osmosis is the diffusion of free water molecules across a cellular membrane down its concentration gradient through channel proteins known as aquaporins, or through simple diffusion until equilibrium is reached. Facilitated diffusion is the movement and diffusion of polar molecules and ions through transport proteins across a cellular membrane. Channel proteins are integral proteins that are embedded in the cellular membrane. They have a hydrophilic channel that allows certain polar molecules and ions to pass through the cellular membrane. Carrier proteins are integral proteins that are embedded in the cellular membrane of a cell. Carrier protein changes their shapes to transport a specific polar molecule or ion across the membrane. Active transport is the movement of molecules against this concentration gradient, which is from an area of a low solute concentration to an area of a high solute concentration with the help of transport proteins. Active transport requires energy from the cell as it takes energy to go against the concentration gradient. Note, equilibrium is never reached in active transport. The sodium-potassium pump is a transport system that exchanges sodium ions for potassium ions across the plasma membrane of animal cells against its concentration gradient. There is a lower sodium ion concentration and a higher potassium ion concentration inside the cell. There is a higher sodium ion concentration and a lower potassium ion concentration in the solution surrounding the cell. The shape of the pump moves 3 sodium ions out of the cell for every 2 potassium ions pumped into the cell. ATP powers the change of the shape by phosphorylation, which is the transfer of a phosphate group. Endocytosis is a process where the cell takes in large molecules by forming vesicles from the plasma membrane. This requires energy, ATP, as it takes energy to change the shape of the plasma membrane and the cytoskeleton of the cell. Phagocytosis is the process where a cell engulfs a particle as the plasma membrane pinches in to form a vesicle. The vesicle will be digested after the vacuole fuses with lysosomes. Pinocytosis is a process where a cell takes in droplets of extracellular fluid and forms a vesicle as the plasma membrane pinches inward. The cell takes in the dissolved molecules. Receptor-mediated endocytosis is a process where the cell acquires bulk quantities of specific substances, also known as ligands. Ligands bind to the embedded proteins with specific receptor sites. Receptors cluster together to form a vesicle as the plasma membrane pinches inward. The cell takes in the ligands as the receptors are then recycled. Exocytosis is the process where the cell secretes certain molecules by the fusion of vesicles with the plasma membrane of the cell. The contents of the vesicles are now on the outside of the cell.